I thought we'd introduce a little bit basic science. We don't get a lot of basic science, and I know certainly the residents were saying how excited they are to hear about what I get up to in the lab. This is a wonderful opportunity to bring you up to speed with what we've been doing. The objectives of today's talk were emailed to you, but sort of in a nutshell, what we're going to do is look at mechanisms of castration resistance, how the insulin-like growth factor, IGF is how it's shortened, probably how most of us know it, fits into the paradigm of castration-resistant prostate cancer. We're going to look at the components of the IGF axis. We're going to look at its role in normal physiology in cancer. And then we'll focus on what role it plays in prostate cancer, what role it plays with the androgen receptor. And finally, my area of research is looking at the binding proteins, which are a component of this axis, and how they might be involved and how we can target these in treatment strategies of the future. So just a quick little reminder of the mechanisms of castration resistance. It's a complex disease. There's multiple facets to it, and we can start with, and I guess this broadly can be divided up into two categories. Those where the cancer's still being driven through the androgen receptor, and this can be through tissue steroidogenesis, which can be from the adrenal, it can be intratumoral. We can still have it going through the androgen receptor, whether that be through amplification or a promiscuous receptor, which will respond to different ligands such as progesterone or estrogens. But we've also got these things over here on the other category, which is signaling through alternate growth factors, such as the IGF receptor. And we've also got these other ones such as increases in coactivators or loss of suppressors of the androgen receptor, and also anti-apoptotic mechanisms, which is really a large area of our research over at the Vancouver Prostate Centre. So looking at treatments that are currently out there, these are the ones that have just arrived on the scene. We've got abiraterone, affects this tissue steroidogenesis. Enzalutamide, we're focusing really on the androgen receptor, its amplification, trying to inhibit it against mechanisms that it's developed to evade our current anti-androgens. We have docetaxel, which is a microtubule inhibitor, so basically just trying to stop the cells from undergoing mitosis, very general, not necessarily just specific for prostate cancer cells. And over at the Prostate Centre, we're looking at sort of these alternative mechanisms. We've got insulin-like growth factor, we're looking at its receptor. We've got novel ways of targeting the androgen receptor, and we've also, obviously, you've heard a lot about heat shock proteins, Castration, and these other sort of anti-apoptotic mechanisms which we're attempting to target as novel strategies. So the insulin-like growth factor axis is made up of its two ligands, which is IGF-1 and IGF-2. There's two receptors, of which the main one is the IGF type 1 receptor, which is the tyrosine kinase receptor. Also, insulin receptor plays a role, and we'll come back to that in a minute. But importantly for my research, there's 6 IGF binding proteins, and for ease of remembering, they've been labelled IGFBP 1 to 6. Now these have sort of another layer of control of IGF and its receptor, and basically they can be inhibitory, they can be enhancing of its action, and in some cases they can act completely independently of the receptor, and it's a little known area of ongoing research. So for those that like pictures, here we have what's going on with the axis. We've got the IGF receptor, which can act through two intracellular substrates, and it can activate the two major pathways of cellular proliferation. So we've got the PI3 kinase AKT pathway, but also it can act through the MAP kinase pathway with the ultimate goal of cellular proliferation, cycle and cell growth. Importantly, the insulin receptor shares a lot of homology with the IGF receptor, and the second type of receptor is not something we're going to talk a lot about today. And obviously this is yet unknown, but certainly separate receptor that the BPs may be acting through. Now, insulin-like growth factor, as like all important proteins that circulate in the blood, doesn't just, or ligands, doesn't just sort of float around by itself, but in fact it's so important, and from an evolutionary perspective it has a binding protein that looks after it while it's in the blood. So approximately 90% of IGF is bound to one of the binding proteins, which is IGFBP3. And what we've found is in prostate cancer, these cells tend to overexpress IGFBP2 and 5. And what we think the reason for that is that this is, as we said, it's another level of regulation. And what it is this can sort of capture the IGF from the IGFBP3 that's passing by, and therefore is more efficiently able to deliver the IGF to its receptor on the surface of the cell. There's also the possibility that IGFBP3 may be working through this as yet unknown receptor. Now, there's a couple of things that are important so far with regards to the IGF receptor and why we've had trouble targeting it and treating it so far. Firstly is that it shares a lot of homology with the insulin receptor, and you can imagine anything that you're doing that might interrupt the insulin receptor is going to have some pretty grave effects on the body as a whole. It's the majority of the homologies in the tyrosine kinase component of the receptor, but about 50% is with the extracellular and the ligand binding domain. So the IGF receptor, importantly, the IGF receptor is ubiquitous, which is another issue. So it's basically on all cells, but of note it's not on hepatocytes, which makes a lot of sense when you consider this is the main site of glucose homeostasis. So the insulin receptor is going to be playing a very large role here, and from a controlling point of view, you wouldn't want too much crosstalk in this area of the body. So the IGF has many roles that hopefully some of us are aware of, and its first known role is that it's mitogenic in a number of cell types. It promotes the growth in the size of cells, so it actually causes hypertrophy, not just hyperplasia. And this is, I guess, particularly important because there is a fair bit of cross-reactivity or interplay between growth hormone and the IGF axis as well. It plays a crucial role in transformation, so this epithelial mesenchymal transformation and the cell's ability to metastasize. And it gets a mention there, it regulates cell adhesion and cell motility. It can protect cells from a variety of apoptotic injuries, and plays an important role in terminal differentiation and longevity of the cell, so it plays a role in aging as well. Now, specifically in prostate cancer, there's been some epidemiological studies looking at, well, what can we tell from patients that have cancers and how may it be affected? And what we've found from 2 large meta-analyses with almost 9,000 patients in the first one is that there does seem to be an association between your risk of cancer and circulating IGF-1 levels. Now, the odds ratio is relatively small, it's 1.38. If you compare patients that are in the highest quintile with those in the lowest and their risk of cancer. We've also looked at all the other IGFBPs, the expression of receptors, and from what we can tell at the moment, there's some studies that say they do, but when you try and put them all together at the moment, there's no evidence that they, in these large studies, are necessarily correlated with cancer risk or not. And I've talked about it affecting invasion and the like. Now, one of the more interesting parts of the IGF receptor is that it does appear to have an intimate relationship with the androgen receptor, which makes a lot of sense when we consider that it is considered one of the main roles of castration resistance, and this may not just be through its completely separate intracellular signaling from the androgen receptor. And these are the studies, I'm just going to summarise them briefly. They were in this review, and the studies that have been done so far looking at this topic. What we found is that IGF receptor signaling can lead to castration-resistant progression in prostate cancers. Androgens can increase the level of the IGF receptor in prostate cancer cells. And what we've also found is the IGF receptor can alter the androgen receptor phosphorylation and its transcriptome. And in fact, IGF receptor signaling itself is actually able to cause translocation of the androgen receptor into the nucleus independent of androgen. And there was a recent study by Carver et al. And Charles Sawyers over at MSK, and basically they looked at, well, how might these two, what were sort of considered quite separate pathways, one's a steroid and one's a receptor tyrosine kinase, how could they be related? And what they found was that, in fact, there does seem to be a lot of interplay and reciprocal feedback between the two pathways. Now, importantly, they looked at, their study was looking at P10 deficient cancers, and that is, a lot of prostate cancers have this this change. They lose P10, which is like a tumour suppressor in a way. And what it does, so if you lose this, you get signalling through PI3 kinase AKT, which is one of probably the most significant, or one of the most significant intracellular the mitogenic pathways. And that can then lead to formation of mTOR or activation of mTOR, which has a negative feedback, as like most things in physiology, on its own receptor and other tyrosine kinase receptors. And what we've found is that constitutive action of other receptor tyrosine kinase receptors is sort of has an inhibitory effect. So basically would normally be very slowly activating the androgen receptor. But once you remove that effect, in fact, what you get is inhibition of the androgen receptor. Now, if you look at how that might, so basically what we see is that, so if you get activation of the pathway down the left, you get inhibition of the right. If you get activation of the androgen receptor, it targets its androgen receptor genes in the nucleus, and that can release this flip protein, which is a phosphatase, which inhibits the action of AKT. So there's quite a strong relationship between these two pathways. Now, if you look at how that might affect cell growth during castration, what you could imagine is that you now remove the stimulation of the androgen receptor because there's no longer androgen around, so you're going to lose activation of these genes, you lose flip, therefore you lose the inhibition on AKT here. So once you lose that inhibition, essentially you have free range now of the downstream activation of these receptor kinase, receptor tyrosine kinase and their pathways. So you'd think, well, that's fantastic. Why don't we all go out now and target the IGF receptor if it seems like this is the most obvious thing that might be affected by castration? And what we've found is that a number of recent phase 3 trials have all had very disappointing results in this regard. And there's been a number of factors that we can look to as might explain why this is the case. Now, when we look at the different ways of targeting the IGF receptor, when we look at tyrosine kinase inhibitors, one of the main problems is that any tyrosine kinase inhibitor that is relatively specific for the IGF receptor will have affects the tyrosine kinase domain, which, as I said, has an 84% homology with the insulin receptor. And what we find is these patients can have quite critical hyperglycemia and hyperinsulinemia. So what we've done is look at, well, can we do specific antibodies which affect the extracellular domain so it's got less cross-reactivity with the insulin receptor. But we've also found that these have failed to work through a number of mechanisms. One, as I mentioned earlier, the insulin-like growth factor axis and growth hormone are intimately related. So if you inhibit one, the other one upregulates. There's a compensatory mechanism, and that will increase the amount of IGF that's circulating, can overcome the antibody inhibition. IGF-2 is able to activate survival signaling via the insulin receptor and its different isoforms. And importantly, if you inhibit one of the if you inhibit the IGF receptor, which is a tyrosine kinase receptor, as I said, so what it does, it's usually got an inhibitory feedback mechanism on other receptor tyrosine kinases, and you remove that, therefore the PDGF and EGF receptors can take over this pro-mitogenic role. There's one more very interesting way that IGF might be involved in prostate cancer, and that is that you look at the role of IGF in the bone microenvironment. If you look at it in normal bones, what you notice is that IGF-1 and IGF-2 are the two most abundant growth factors that are stored in skeletal bone. It's involved in bone resorption and formation, and this is going on normally. And what we find is that IGF-1 can upregulate the expression of both the receptor activator of nuclear factor kappa beta, RANK, and its ligand, and it has a role in osteoblasts that bind to RANK. And what we found is that castration itself increases IGFBP5 expression in the bone microenvironment. Now this is really, I guess, really plays into this notion of a metastatic niche that gets set up in the bone. So basically the bones are primed for prostate cancers that may be particularly receptive, particularly in castration, in the state of castration, to accept any sort of circulating tumour cells that may want to land in that environment. And what's the evidence for that? So what we've got is this study by Zhu, and he showed that IGFBP5, so the binding protein 5, is particularly, so this is in normal bone, is expressed in osteoblasts in the endothelial. So basically it's got the landing site in the endothelium, and it's also got the osteoblasts which are going to be involved in the bone formation. And what they found was that in a microarray analysis of human bone marrow stromal cells after castration, that 159 genes were upregulated, 84 were downregulated, and of the 159 genes that were upregulated, IGFBP5 was that which was most consistently upregulated. When you look at immortalized human marrow stromal cells and their expression of IGFBP5, after castration, it increases almost 20 to 30-fold, and this can be rescued, so either you can bring those levels back down to normal or even better with the addition of R1881, which is an androgen which we use in the lab. So it's essentially testosterone for our purposes. So as you can see, so this is without any rescue, you get quite, 2.5-fold induction, but once you add in the androgen, it returns back to normal. So now we're going to look at, well, what role are the IGFBPs playing in prostate cancer? And this is what we already know. We know that the IGFBPs increase after castration, that IGFBP2 and 5 increase, and IGFBP3 appears to decrease as you move from low to high-grade cancers. And in one study, as I said, but when we look at multiple, it doesn't always play out, but there is some evidence that IGFBP2 is an independent predictor of PSA failure, and this is really looking at tissue specimens as distinct from circulating serum levels, which the population studies have really looked at. And now we'll turn now to work that's been done here at the Prostate Centre with Dr. Gleave over the last 10 years, looking at these BPs and their differential expression with time and castration. And this is a study that was done that showed the change in the IGFBPs in a Ischia model over time after castration. Now, Ischia is a mouse model, but the tumour that grows is androgen dependent, and most importantly, the androgen receptor in the Ischia tumour, it's a wild type receptor. Now, it's relatively basically, I guess, when we look at the typical cell lines you'll have heard about in prostate cancer, whether it be LNCAP, DU145, PC3, well, certainly the LNCAP has a mutated receptor, which makes it very promiscuous, so it can respond to a number of different ligands. DU145 is very promiscuous, and PC3 doesn't have an androgen receptor at all, in fact, and really behaves like a sort of a self-driven androgen-independent cell line. But importantly, in this Ischia with a wild-type androgen receptor, once you castrate, you can see after 28 days you get an increase in IGFBP2 levels. IGFBP3 decreases after castration, but in fact after a month levels will return to normal. But most importantly, you see that IGFBP5 essentially is non-existent in wild-type tumours before castration, but after castration, IGFBP5 is very quickly and very highly upregulated. We also saw that was in vivo. Now when we look at LNCAP cells, this is an in vitro model. Once again, you've got in the presence of androgen, there's really no change in IGFBP2 levels over time. But in the absence, this is castration, sort of an in vitro model of castration. You can see that after sort of 24 or 48 hours, you've got increasing amounts of IGFBP2, and by 72 hours, you've got very highly upregulated IGFBP2 in these cells. When we look at that effect in LNCAP xenografts, so we're moving from the lab into a mouse model, once again we see by about 10 days IGFBP2 levels are increasing, and by 3 to 4 weeks they're highly upregulated. There's also, in this particular paper, they looked at a tissue microarray of the various stages of castration, and what we found was that in benign tissue, the levels of IGFBP2 are very low, but as you move through primary cancer, it's increased, which corresponds to B, and as the amount of castration increases, the expression of IGFBP2 by immunohistochemical scoring system increases. Now, I guess one thing we like to do in the lab is, it's one thing to show that these things, if you inhibit them, then yes, or change the castration levels, that you can get a change in the growth. But one way that we sort of try and work out what role a gene or a protein is playing is by overexpressing it or knocking it down. So when you look at overexpressing IGFBP2, what we can see is that cells, this is in a xenograft model of LNCAP, so your LNCAP cells, get them to overexpress that particular gene. These cells grow quicker, and certainly after castration, they really take off and grow a lot quicker than wild-type cells. And this was both with tumour volume and PSA values in these mice. We've been able to do that for IGFBP5 as well, so we've overexpressed it in certain Allen-cap cells. And as you can see here, 2 subclones that overexpressed BP5 grew a lot faster after castration compared with those that were not. And that's the same with PSA. So you can see they're castrated here at time 0, PSA drops, but those cells that express BP5 or overexpress it at great amounts, they take off and grow a lot quicker. Now, another part of that study that was just done, and this is a very interesting graph which I'll just walk you through slowly because it looks very complicated, is what is this role between IGF receptor and testosterone. So you can see they've got a number of experimental conditions, whether they've got testosterone or not, we've got IGF or not, and there's an antibody. But just to simplify it a little bit, we'll get rid of a couple of lines because they were sort of plasmid control and wild types and 2 different sublines. So if we just look at this, the first thing to notice is the difference between the left column and the right one in this box is that we've added IGF. Okay, and as you can see, there's not a big change in either the Allen-cap wild-type cells or those that are overexpressing BP5. Now, what is important to note that the BP5 grows at a greater rate than the wild-type cells. But now if we go over here, so now we have testosterone. So that was in castrate conditions, sorry, on the first in the first box. Now in this box, we've added testosterone, and if you look from going from IGF minus to the addition of IGF, you can see there's quite a significant increase in the growth now of the cells that are the wild-type cells. But in those cells that were BP5 overexpressing, there is an increase compared to where they were before, but there is no difference, once again, between the addition or absence of IGF-BP5. And what that means is that IGF-BP5 is already essentially maximally stimulating, is what we've inferred from this, the actions of the IGF axis in these cells. And one more thing to note is that in fact the addition of the IGF brings up the wild-type cells up to approximately the same level now as those that are overexpressing BP5. So BP... What's that, sorry? Oh.
It's the difference between... It's just the difference between DHT.
Yes. Oh, yes, I see. Yeah, but when the DHT is present, it brings it up to... Sorry, you're right. Brings it up to maximal stimulation. And this really brings me to where sort of I'm going to start to come into with my research, and that is the role of the antisense drugs that Dr. Gleave is developing and been involved with for approximately 10 years now, and the latest and the sort of next generation antisense technology that we have. It's just a very quick reminder for those that hear a lot about antisense but don't know how it works. Under normal conditions, we've obviously got DNA, we have transcription occur. So you get your message, the introns are spliced out, and then the message heads off into the cytoplasm, ribosomes attach and start translating protein. Now what the antisense does is, this is a single strand DNA, it's a single strand DNA oligonucleotide which has a complementary sequence to that of the message that you're intending to target. And so what it is, so remembering that you know, by the Watson-Crick sort of associations, you have cytosine and thymine and adenine, adenine and thymine, cytosine and guanine will match up. And what we find is that if you can design this so it specifically will target a certain area of the message, then they will bind, and then it can either attract enzymes such as RNase H, which can degrade this complex, or in fact, just by the fact that it's there, will prevent translation of the protein. So we've looked at this with just targeting IGFBP2 or 5 already. And the most important thing to consider when you're talking about antisense actions is that they have to be sequence specific. So we always have what the sequence we're interested in, and we always compare it to a sequence which has a few nucleotide mismatch or a scramble, and it has to be dose dependent. Because what we find, these antisenses are quite toxic within themselves, and that is that they can sort of bind all sorts of proteins that are floating around the cytoplasm. This was certainly the case in the early generation antisenses, and in fact they had quite a good inhibitory effect just by themselves. And so looking at an IGFBP2 antisense which is from, is it now, almost 10 years ago. So we get a decrease in the cell viability with increasing concentrations, and as you can see in the mismatch control, there's no change in the viability. When you look at the growth of Allen cap xenografts, those that are treated with the Antisense grow a lot slower than the wild type, and that's for serum PSA, and tumour volume. And in fact, looking at the specificity, this is just a sort of typical tumours. Those that were treated with the Antisense have a decrease in the amount of IGFBP2 message compared to those that were treated with the mismatch control in this study. Looking at an Antisense now that targets IGFBP5 in the Shionogi tumours. Remember, those were the tumours that grew, very highly overexpressed BP5 after castration, and you can see that you get slower growth of tumours in this model compared to those that are treated with a mismatch. OGX-225 is a bispecific, it's a second generation antisense. And what it does, it targets both IGFBP2 and IGFBP5, and was developed, I'm not sure now, 10 years ago was it? But someone did this particular sort of analysis where they looked at the sequences of both IGFBP2 and 5, and they found several areas of homology. And what they found was, so this is not linking, this is just showing nucleotides that are the same. So within BP5 and 2, there's this which is 22 nucleotides, and there's one here 17 and one that's 15 nucleotides. And what they did was develop OGX-225, which covered these 20 nucleotides so that it would hit BP2 and 5 simultaneously. Now, it also happens that it has 100 homolysis, or has 100% hit on IGFBP3 and transforming growth factor beta 1. There's only been one study that's previously published looking at OGX-225, and that was done by Alan So a few years ago, and he looked at its effect in breast cancer. He started by looking at the expression of IGFBP2 in a breast cancer tissue microarray, and found that while 95% of benign cancers didn't express any BP2, this level was relatively low in benign tissue, was 95% did not express BP. In fact, that was only 20%, and the vast majority of breast cancers do express BP2, which would make it appear a good target for some sort of antisense technology. And so what he did was he took a breast cancer cell line which overexpressed BP2, and what he found was this sequence-specific and dose-dependent reduction in viability, or in this case, we started by looking at reduction in the protein. He then looked at a decrease in the viability, and he went on to do a xenograft study which indeed showed that treatment with OGX-225 decreased the growth of tumours in his mouse model. Now I've come in, and my job is to start looking at OGX-225 in prostate cancer, and in particular sort of MDV resistant or enzalutamide resistant cell lines. So when we started, we looked at RNA-seq data for IGFBP2 and 5 across a range of prostate cancer cell lines, and we compared them to Allen cap. And what we have here is some castration-resistant cell lines that we've developed. As you can see, IGFBP2 and 5 increased 3 to 4-fold in these cells. If we look at how that effect looks in a Western blot, we have a benign cell line where you can see the expression of BP2 is relatively low. When we move to a castrate-sensitive cell line, which is, an androgen-sensitive cell line, such as Allen cap, BP2 expression increases. And when we look at castrate-resistant cell lines, such as V16A or 22RV1, we can see the level of IGFBP2 has increased again. We also looked at IGFBP5, but at the moment we're having some troubles with the antibody, and we won't look at that too much just at the moment until we get that sorted out. Now this is some preliminary work that was done by Moto Muramaki, who was here a few years ago, and what he found was in Allen cap and PC3 cells, OGX-225 was able to decrease the expression of their respective BPs. So Allen cap only expresses BP2 for all intents and purposes, so he was able to decrease the expression of that both at the message level, this is a northern blot, and at the protein level. And PC3 expresses BP5 but not 2, and he was able to show that he was able to decrease the expression of both the message and the protein. He did some very nice experiments in vitro showing that in fact you can decrease the viability of these cells for our PC3 and LNCAP, and this was largely through an increase in the apoptotic fraction. He did some xenograft studies, which were very nice, again showing that in LNCAP xenografts he was able to delay the time to castration resistance or delay progression with treatment by OGX-225 compared to a mismatched control or a control oligonucleotide. And this was true for tumour volume and PSA. And obviously it was specific with IGFBP2 message being knocked down, not so for the mismatched controls. This is also the case in PC3 cells where we're targeting IGFBP5, we get a delay in the growth. I mean, these are already androgen independent, so we're not looking at progression, we're just looking at the growth time of the tumour. Now looking at castration resistant cell line that we've developed, as you can see we've got OGX-225, where it knocks down. I mean, we're not able to show a dose dependent just yet, but we're now performing an experiment looking at lower values, but you can see they're exquisitely sensitive to knock down with OGX-225. Yeah. So V16D is a castrate resistant cell line that was developed by Hide Miyake. So what he did, we took LNCAP cells, grew them up castrated, so the LNCAP regress, and then they come back as a castration resistant cell lines, and we took them off at that stage, and we've got several which we call 16, and then some cells were put back in, and then they grew up in a castrate environment, and then MDV was added, and that's where we're going to get our MDV resistant cell lines, so cells that grew up in an MDV environment and in fact became resistant to that, so our enzalutamide resistant cells, which we'll talk about in just a minute. So we've looked at the viability of this castrate resistant cell line with OGX-225. You can see it's exquisitely sensitive to treatment with OGX-225. We've also got 22RV1 cells, which are another castrate resistant cell line. And in fact, we've done a synergy assay looking at the effect of OGX-225 with MDV, because I think this is the next sort of paradigm in treatment, which is, you know, we really want to take castrate cancers and we're going to start treating them with MDV. And is there anything that can prolong the effect of MDV? So this is a synergy assay where you treat cells with MDV alone, with OGX-225 alone, and then in combination you hope to see a synergistic effect. Using a Calcusin, which is a program which can tell you whether there was a synergistic effect or not. The key here is this graph in the bottom right corner, and what it shows is that if it's less than 1, it's a synergistic effect. If it's more than 1, it's antagonistic. So we can see at the lower concentrations, which is as you'd expect here, we get a synergistic effect between OGX-225 and MDV, but at the higher concentrations or maximum cell death, basically most of the work's been done by OGX-225 in this particular combination that we used. So that really brings us to the enzalutamide resistant cell lines, the final bit of the talk, and we're looking at, so this is going to be the next problem we're going to have in prostate cancer. What happens when prostate cancers are resistant to abiraterone, enzalutamide? I mean, we've still got chemotherapy, but you know, what other options can we offer these people? And that's what we're looking at. Can we find a way that, you know, will treating the BPs in this particular environment make any difference? So the first thing we did was a microarray gene analysis, and which looks at the gene expression profile of these cancers. And while this is very complex, the most important thing to take away is you can see that IGFBP5, which is this line here, sort of flat, but it really does spike up in these 42D cell line, which is one of our enzalutamide resistant cell lines. So if we look at that sort of in a more clearly, you can see there's almost a 12-fold induction compared to the base cell line that was used in this case. Now, next generation sequencing is a term that you're going to hear more and more, and this really refers to it's a whole transcriptome analysis. So what we're looking at, what is all the message RNA within a cell, and because the difference is with a microarray, you really need to probe for the genes that you're interested in. So you need to know what you're looking for before you start looking. Whereas with this RNA-seq, which is the next generation, in fact what you do is you get a readout of just all the message that's in the cell, and then it takes a couple of months of pretty heavy computing, and they can then go back and work out, well, which genes were being expressed. So using this technology, we've performed this analysis on a number of our cell lines, comparing to LNCap, which was set as the base in this case. You can see with our castrate resistant cell lines there is an increase in BP2 and 5. As we mentioned previously, but importantly, when we get into these enzalutamide resistant cell lines, in fact, the overexpression now, or the increased expression of IGFBP is very marked. Okay, we're getting up sort of 350-fold expression in this particular cell line. Now that's not quite the case for IGFBP2. It's in the range of 2 to 3, but we tend to see this. IGFBP2 is not expressed to sort of quite the degree. But the next issue is, well, that's fine to have from this RNA-seq data. Does that translate to actual message in the cells that we have by PCR? And we find that, yes, once again, by PCR, we've got marked overexpression of the message in this particular cell line. But also, there is quite a big increase in the other cell lines that we use over there in the lab. One important thing, IGFBP2, so does this message correlate to protein? Well, it doesn't appear to at this stage insofar as the amount of IGFBP2 goes. Remembering though that it's already very highly overexpressed when you moved into these castrate-resistant cell lines from both benign tissue and primary cancers. So using OGX-225, we're able to knock down the expression of IGFBP2 in these 42D enzalutamide-resistant cells. We're able to decrease the viability. You're treating them with OGX-225 in both the 42D and our 49F, which is our prototypical enzalutamide-resistant cell line that produces PSA. And we can see that most of this effect is occurring through apoptosis with an increase in the apoptotic fraction of these cells, both for 42D and 49F. And really, and the final step in any scientist's goal to show that indeed this is occurring through apoptosis is to show that there's an increase in the amount of part cleavage, which is one of the components of the apoptotic pathway. So that's, you'll be relieved to hear, we've come to the end of talking about OGX-225. So in summary, IGFBPs are increased in castration-resistant prostate cancer. Targeting the BPs with Antisense reduces the viability of prostate cancer cells, both in vitro and in vivo. It causes this cell death through an increase in apoptosis. It can delay the progression to castration-resistant prostate cancer in vitro and in vivo. And importantly, OGX-225, which is the new bispecific antisense with the sort of the second-generation technology backbone and stability, is able to target both 2 and 5 and has good in vitro effect at this stage. I guess the last thing to mention, so there's ongoing, we've got the xenograft studies going, so we've got 42D, 49F cells growing in mice at the moment, being treated with OGX-225 or scramble. And we've also got V16D, so a castration-resistant cell line growing in mice, being treated with 225 or MDV or a combination of both to see are we able to get a synergistic effect which would have some clinical relevance for us obviously in the clinic. So thank you very much.